It's really a very special and personal pleasure and privilege for me to introduce this morning's Grand Rounds guest speaker, Allan Templeton of Washington University. He received his undergraduate degree at Washington University, his master's and doctorate at the University of Michigan in zoology, statistics, and human genetics. He rose through the academic ranks and is now the Reb Stock professor at Washington University in biology and also in engineering. He's had many international awards at leading universities, such as the University of Oxford and others. He has several hundred publications in leading journals, has been on editorial board, and has had presidential or officer positions in major scientific societies in areas of conservation biology, genetics, and evolution. He, for example, won the David Murdoch Dole Award in Human Genetics in Stockholm in 2002, and he was listed in the top 1% of cited authors for life sciences journals in the year 2005. In terms of his contributions, they cover a very broad array. It would be, if I were just to say what he hasn't done, I could stop right now because he's covered so many different areas in his repertoire of scientific accomplishments. In a brilliant career spanning now 3 decades, he's made major theoretical, practical, and health-related contributions in a dazzling array of areas of biology and life sciences. He's a field biologist in the tradition of Darwin, actually an adventurer, more like in the traditional movie figure of Indiana Jones. Field biological work led him to discoveries cutting across many species, from salamanders to gazelles to flies and to humans. He's also a conservation biologist and personally worked to save some endangered species on the planet, understand the interaction with the ecosystem, and extended his interests to the interaction of life with entire landscapes. As an evolutionary biologist, he's applied his experience and skills to human evolutionary and population genetics problems. He's a world-renowned population geneticist, invented and applied novel analytical tools for genetic analysis, which have become part of textbook knowledge. Indeed, Allan has released the first edition of what will surely become the standard textbook in population genetics, written in a very clear manner, and hopefully it'll be translated into Hebrew at some time as well. To focus on human genetics, he has done landmark research on the evolution of HIV virus and the development of AIDS in human populations, work on the genetic epidemiology of coronary artery disease, and its implications for other complex phenotypes. Beyond his research work, he's a highly sought-after educator with numerous international invitations to run courses, supervise action programs in population biology, and so forth. He's been a keynote speaker at some of the most important international symposia. My own personal friendship with Professor Templeton and his wife, Dr. Bonnie Templeton, began approximately 7 years ago when he was invited here as a visiting professor at the Technion. Since then, we've cultivated a scientific, professional, and most importantly, personal friendship from which I draw great, and others here draw great inspiration. He's a warm and caring individual who has coined and lives by the motto, people first. He's currently in Israel as the Fulbright Distinguished Visiting Professor, and we're delighted that he's agreed to give his Grand Rounds talk this morning on the topic, Ha'im Tam Ha'evolutzia Enoshit, has human evolution ended? Let's hope not, we'll find out, but if it has, then Allan is surely its pinnacle. Thank you.
Now I got to live up to that introduction. Yeah, Shraga asked me to talk about this topic, is human evolution stopped? And actually many people feel the answer to this question is yes. And the reason is they say, well, humans really no longer adapt to their environment by natural selection. They adapt to their environment using their intelligence and their culture. So cultural evolution is occurring, but biological evolution no longer exists in our species. And in fact, if you look at the literature, there's some support for this idea that things like natural selection are no longer as important. So this is some work done by some colleagues of mine, Jim Chevrud at Washington University, and Becky Ackerman was a graduate student at the time. And they looked at human fossils over the last 2.5 million years. They got virtually every fossil find they could and made measurements on it. Now, I'm not going to go into the math because Jim is very mathematical, but if you make a lot of measurements and look at how they're correlated and how those correlations change, and you can study this in current modern humans, chimps, and gorillas, and it turns out it doesn't make any difference which model you use because they're so similar. You can look at how, when traits change, do they change in the way that you would expect just given their developmental constraints? Or do they evolve in a way that breaks down or changes those correlations? And when that happens, that's evidence for natural selection. And what Jim and Becky found was that starting with the common Australopithecine ancestor, it's split into two lineages, one which we call the robust Australopithecines over here, and the other, the gracile, which eventually led to modern humans. And what they showed on these arrows is, by the intensity of the darkness, is the evidence for selection, shaping the face, jaws, and teeth. And what they found was, going into the robust Australopithecine line, selection was getting more and more intense. So these very derived, robust Australopithecines, their face, jaws, and teeth had been strongly shaped by natural selection. On the other hand, they found that in the gracile, the selection gradually reduced. And by the time they got here, the time of Homo erectus, they could detect no selection in the human lineage at all for our face, teeth, or jaws. They were no longer subject to natural selection, and they concluded that already 1.9 million years ago, Homo erectus had evolved a culture and a tool-making kit that no longer permitted humans to adapt to their dietary environment through their teeth and jaws. We now did it through our tools and our culture. So that's the basic idea. But what I would like to first point out is that although Ackerman and Cheverud rejected the adaptive evolution of the human face and jaws over the last almost 2 million years, their null model was not no evolution, it was what we call neutral evolution. And this leads to what I think is the first misconception people have when they say, has human evolution stopped? If we look at the fossil record and say, has the human face stopped evolving? Well, when you look at the beginning of this 1.9 million period, here's a face and teeth and jaws, very different from what we have today or just in the recent past. Obviously, the face, teeth, and jaws have evolved, have changed over the last 1.9 million years. Yet Cheverud and Ackerman say there's no natural selection on these traits. So how can you have evolution without natural selection? And that really leads to the major mistake, one of two major mistakes in this argument, is human evolution stopped. Many people just equate evolution to adaptation through natural selection. But in fact, that's not our definition of evolution. Evolution is simply a change in the type or frequencies of genes or gene combinations over the generations in the gene pool, where the gene pool is the set of genes that a population collectively shares. It has nothing to do with natural selection per se. It's just genetic change at the population level over time. And there are many factors that can cause evolutionary change, and these are called evolutionary forces. And of course, one of the major ones is indeed natural selection. Natural selection is a major evolutionary force, and no one denies that. And it favors those traits that allow us to live, find mates, and reproduce in the context of a particular environment. But there are many other evolutionary forces. Mutation, the ultimate source of all genetic variation, is also an evolutionary force. It can change the gene pool. Another one is just genetic drift. All populations are finite. And as any experimentalist knows, your results can vary from replication to replication just by sampling error. And this sampling error is actually a very powerful evolutionary force that's called genetic drift. And of course, another evolutionary force is gene flow. Populations have an existence over space, and they can spread genes through space. And I'll give specific examples later. This can be a very powerful evolutionary force. And the most relevant one for the problem of the Cheverud and Ackerman study was developmental constraints. If you select on one trait, even a trait that is not being subject to natural selection will often evolve. Why? Because genes don't work in isolation. You don't have a gene just for this molar. That's not how genes work. They work through developmental processes, and when you change one trait, you will always change others. And in fact, in human evolution, Cheverud and Ackerman agreed with everybody else, there had been selection for a larger brain. And when you select for a larger brain, guess what? You also simultaneously will change the face, teeth, and jaws as a developmental constraint. So in fact, if we want to understand where the human face came, it turns out that it did evolve, but it evolved exactly along the lines that we would expect for a primate. Humans achieve their big brain, I'll show you later, by retaining fetal growth patterns longer than other primates. And as a result, our human adult face is more like that of a newborn primate. So here you can see a newborn chimpanzee with a very flat face, a very human-like face, particularly compared to the adult chimpanzee. So we don't grow into that. We stop at this point, basically. So in fact, the human face has evolved, but it has not evolved through natural selection. It has evolved as a developmental constraint for a big brain. Now, there's actually an implication of this, for some of you at least, and that is it turns out our teeth, our jaws get relatively small as our brain gets bigger, and so do our teeth, but the teeth get smaller at a slower rate, which means our teeth are too large for our jaws. And for example, if you look, I got a crooked tooth here. Why? Because I'm the typical human. My teeth are too large for my jaws. And I'm sure some of you in this audience make your livelihood out of this evolutionary fact. Now, another thing is that even when evolution and natural selection is not operating today, its operation in the past can have a dramatic impact on the traits we see today, and in particular, how these traits are changing. Remember, all traits are gene by environment interactions. So if you had a trait that evolved in the past, but now the environment has changed, you will change the nature of what traits those genes code for and what they do. Now, for example, one trait that has changed quite a bit in the United States is obesity. So as we go from 1991 to 2004, there's been a dramatic increase in the rates of obesity in the United States. Now, nobody's saying that we've evolved fat genes over this time period. This is not evolution. We can explain this by our shift in our diet, where when you look at the diet from here 1970 and here 2000, the main difference is actually not so much what we're eating, but how much we're eating in the United States. We've just increased the amount that the average person eats substantially over this time period. So no one is saying this obesity epidemic is related to evolutionary change. Rather, it's due to a change in an environment. But remember, the genes that lead to obesity were selected under past environments, and now they're in a new environment with a much greater calorie input, and one of the responses is obesity. And this in turn has all sorts of secondary consequences, one of the most dramatic being that we're also engaging in an epidemic of type 2 diabetes. Now, long ago, James Neal proposed a very interesting hypothesis, talking about how past evolution by natural selection would affect current changes in traits. And his hypothesis was called the Thrifty Genotype hypothesis. And the basic idea was that past selection could be very intense, and therefore an allele could be favored and go to high frequencies under these past environmental circumstances. Now culture occurs and makes the trait neutral. It's no longer under natural selection, but culture also changes the environment and therefore changes the manifestation of how these genes translate into traits. And so new traits appear that were never really part of the original evolutionary process. In particular, Neal proposed that many human populations in their history had gone under very severe and periodic famines, and these would select for what he called thrifty genotypes. But now, when the environment includes abundant food and high-calorie diets, as you saw we now have, these same thrifty genotypes, Neal proposed, would be predisposed to type 2 diabetes and obesity. Now, these effects are late age of onset, and to an evolutionary biologist, they don't have much of a reproductive fitness impact. So these are neutral traits. They can be very clinically significant, but they're not really evolutionarily significant. Now, he looked at several populations to support this, and Neal's original studies were based more on Native Americans. So, for example, the Pima Indians in the American Southwest had a historical record of very severe famines that killed off a large proportion of their population. And now when they were put into a high-calorie environment today, 50% of the adults developed type 2 diabetes. Similarly, Hispanics had a somewhat similar origin, but it wasn't just all Native Americans. Another Native American population that had a stable agricultural system for thousands of years in an area that was not prone to periodic famines doesn't show this huge increase in diabetes. And since then, they've done many other studies. So populations without a history of periodic famine have lower rates of diabetes than populations that have this history. Here was a case where we could see the change in time. This was an island in the Pacific that had very poor diet. Once again, a record of many famines. But then after World War II, guano mining started on the island. The islanders became rich. They changed their diet. And the rate of diabetes went from 0 to 41% of the adult population. You see a similar thing in New Guinea and Australian Aborigines, people that also had a history of famines. When they're put into a context where their diet and exercise levels change, huge amounts of diabetes. Now, this hypothesis has been around, I say, since '62, but it's really been revived with molecular genetics. And I'll just give one study, but there's now many that support this thrifty genotype hypothesis. First of all, there's several studies that have indicated specific genes that are associated with increased risk for type 2 diabetes. And one is in the calpain-10 gene. It's called SNP, the single nucleotide polymorphism 44. So this is a study where they looked at genetic variation around this. So when you're conditioned, the fact that you're starting with a variant, you know you have a peak. So basically, things up here are indicating higher levels of variation. And as you look around the region, you see the variation drop because you're no longer conditioning on a known variant, but it's still relatively high. Now, one signature of natural selection, when selection takes an allele and makes it go to high frequency very rapidly, is what we call a selective sweep. The genetic variation near that allele is eliminated. And so they looked at several populations, ones that were not subject to periodic famines in their history versus one that had a lot, and this is kind of a gradient. And as you can see, when we go to populations with the increased history of periodic famines, we see the selective sweep. The genetic variation is just gone away. So they found that, in fact, Neal's hypothesis was right. The genes that predispose us for type 2 diabetes are in high frequency in populations that have periodic famines in their recent evolutionary history. And that frequency is high because of past natural selection. So the past natural selection has a big impact on the changing phenotypes we see in modern humans. Now, it turns out people have really liked this idea, so now there's a thrifty genotype variant for virtually every systemic disease in humans. And I'll just give one more that relates to cardiovascular disease. As I mentioned earlier, humans have been selected for a large brain size, and we've seen dramatic evolution in that over the last 2 million years. And this brain size has been achieved developmentally in a very interesting way. Here is a plot of brain weight versus body weight, and birth occurs here. Now, up at this point, this would be the same for humans and chimps. As I mentioned, chimps and humans are born with virtually the identical brain-to-body ratio. But then what happens in chimps is that very rapid growth goes into a slow mode. Okay? And so the adult chimp does not have the large brain of a human. What humans do is we maintain that fetal growth pattern, that fetal growth rate for another year after birth. And then we get the typical primate adolescent to adult growth pattern. And that puts us in a very different range for our adult size. So humans are very unique among the primates in having this extended postnatal brain growth period. Now, if you look at a newborn human and a newborn chimp, what's the most dramatic difference you see between them? It's not their brain-body ratio. They're virtually identical. It's what we see. Human babies are fat. And people don't realize how unique that is. Of all the primates, humans are the fattest. And in fact, of almost all mammals, human babies are the fattest, the only exception being sea mammals, which use subcutaneous fat for insulation. Other than that, we are fat. That is the hallmark of our species that distinguishes it more from virtually any other terrestrial mammal. And why are we born fat? Well, if you know about it, fat is both a good energy source, but also there's structural components of fat. We need this fat to sustain that brain growth after birth. And in fact, if you have a baby who is born that's not fat, as many of you know, that's a warning signal for lots of problems. We need that fat because we have to sustain this very energetically expensive structure in this rapid phase of growth for the first year of life after birth. And this has resulted in reshaping many of our genes involved in lipid metabolism, for example. And this is the area I work in. I'm not going to give the details. That would literally be a whole nother seminar. But what we find is that the same genes that have been selected to meet this unique demand for humans to get their large brain in the modern environment with our modern diets and so on, these are the same genes that predispose us to cardiovascular and Alzheimer's disease. So once again, the evolutionary past keeps haunting us in the present. And even though we're seeing rapid changes that are not evolutionary, we in fact, to understand why we're seeing an epidemic of diabetes and so on, we have to understand its evolutionary history. Now, I mentioned several evolutionary forces, and one was gene flow. And this is certainly a force that is undergoing rapid change in the world today. Over the last 1.5 million years, gene flow has been dominated by a pattern that we call isolation by distance. That is, most humans mated with nearby humans. Okay. Nothing very unusual about that. A gene could spread throughout our whole species, though, by using each population as a stepping stone, because genes can replicate copies of themselves. So they can take many generations. So even though most dispersal of humans was very limited, genes could spread throughout all of our species. Now, this model of stepping stones leads to a very explicit prediction. There should be a very strong correlation between genetic distance, that is a way of measuring how distinct two gene pools are, and geographical distance. And in fact, this is a recent study based on several hundreds of microsatellite loci, and they just plotted against geographical distance. Here's the genetic distance. And as you can see, geographical distance is by far the single most best predictor of how distinct any two human populations are going to be. Not skin color, not all the attributes we use to classify people into races. They don't work. Geographical distance is by far the most important factor, reflecting this evolutionary history of a gene flow pattern of isolation by distance. But that isolation by distance pattern has certainly been greatly altered starting about 5 centuries ago, but particularly in the last 50 years. We now live in a very mobile world. And that's bringing together formerly distant people. And as a result, currently humans are undergoing a dramatic evolutionary process that's reducing the genetic differences among human populations due to this increased gene flow. And here's just one example. Due to various policies we won't go into, Europeans and Africans were brought together in the North American continent. And once again, these distinct peoples began to interbreed. And currently, African Americans represent this gene flow between Europeans and Africans. And this shows how the evolutionary changes occur. So here's a particular allele at the Rh blood group locus that's in low frequency in Western Europeans, high frequency in Western Africans. And you can see it's intermediate in African Americans. So that's what gene flow does. It tends to homogenize, reduce the differences between populations. And this is an evolutionary process that has occurred just in the last couple of centuries and is still going on, in fact, going on at a higher and higher rate, not only in America, but all over the world. Now, this bringing together of formerly distant peoples is making us a highly dynamic species right now. And in particular, it is reducing the genetic differences between our populations while simultaneously increasing the genetic variation that we have within populations and individual levels of heterozygosity. So if we look at what happens to genetic diversity, it is split between genetic diversity within local populations versus between. And the primary balance of how you shift genetic diversity is this genetic drift, this sampling error I talked about, which tends to decrease local variation and increase differences, versus gene flow, which does just the opposite. It decreases this, increases that. And now we've had a very rapid process because we've increased gene flow, and by increasing our population size, we've decreased drift. So humans currently are undergoing a rapid evolutionary change of converting genetic variation between populations into genetic variation within, where it can also increase the level of heterozygosity. So we are undergoing a rapid evolution right now. Not no evolution, but very rapid evolution. And this has significance. Carl mentioned I work with lots of critters. One of them is a Spix gazelle, the only gazelle with an inflatable nose. We won't go into that. But we were involved in a program with Spix gazelle where we had inbreeding. That is, because of a small founder size, there was a lot of inbreeding. And we found that as the level of inbreeding increased, the viability of the animals decreased. And this is called inbreeding depression. And it also occurs in humans. In fact, the rate of change in the Spix gazelle is statistically not distinguishable from the rate of loss of viability in humans for inbreeding as well. So we know this increased homozygosity can have clinical significance. Now, there's recently been many studies on humans documenting that, in fact, that we have these homozygous segments. And of course, we know some people that have inbreeding, but this was a study where they picked people without known inbreeding. But because we have this evolutionary legacy of isolation by distance, in one sense, we're all inbred to a certain degree. That's what it means. And so by looking at a person that had no known history of inbreeding, as they scanned the chromosome, here where they're measuring the copy number, they showed, yeah, there's no deletions or insertions on this segment of the chromosome. This is on chromosome 4. But as they went through the person, bang, they got an area here where there was complete homozygosity. And this is reflecting one of these common ancestors that we don't know about many years ago. All of us have these homozygous segments in our genome, virtually every one of us. But what happens with the current gene flow patterns is that these segments are becoming rarer and smaller at a very dramatic rate. So we are seeing dramatic molecular evolution generation by generation in our human species. And this does have clinical significance. There was a recent study that looked at people in Croatia. So for example, they looked in a village and they said, well, if you are from the same village all 4 of your grandparents were from, we'll call that endogamous, and they had different levels. So they looked at Croatians with similar diets, environments, socioeconomic status, but that had been differentially affected by these new gene flow patterns, with this group being the most outcrossed, influenced by the modern transportation the most. And sure enough, they found that heterozygosity levels increased as you went from the more traditional breeding system to the newer breeding system that we now have. And then they looked at several traits, and they found many significant effects with this increased heterozygosity. So for example, systolic and diastolic blood pressure, cholesterol level, LDL cholesterol level. In every case where they got a significant effect, it was a beneficial effect to have higher heterozygosity. So this is an evolutionary process that is occurring rapidly right now at a global scale in humans, and this is one that hopefully will actually have some benefit to us. The argument that we started with was that humans now adapt to their environment by using their intelligence and culture. There's another flaw in this argument, not just that evolution is more than just natural selection. And the second problem is natural selection is still here. It still is operating on human populations. So although we kind of like to think that our culture completely buffers us from the environment, it does not. We interact with our environment in many ways that can induce very powerful natural selective forces. And I'll just use one example, HIV, the retrovirus associated with AIDS in humans. Okay, we know that HIV can induce high rates of mortality in humans through the development of AIDS. We also know, and my work and others has shown this, there is tremendous variation in the rate at which AIDS develops after HIV infection in humans. And some people who have been HIV positive literally for decades don't show the slightest sign of progressing to AIDS. And so for HIV to have an impact on adaptive human evolution, this variation in response to HIV infection must be inherited, and the genotypes must have different reproductive fitnesses when exposed to HIV. So we also can do, once again, identify specific genes that are involved in this resistance to HIV. And here's just one genome scan that indicated a position that corresponded to the candidate locus CCR5. And this is a very good candidate because HIV-1 initially interacts with cell surface receptors, primarily CD4, to gain entrance to the cells. Once it binds, we undergo shifts both in the HIV and the human cell that allows it to bind to the CCR5. This is a human protein. And that is how HIV gets entry into our human cells initially. Now, HIV is very clever. It can evolve its use of these receptor proteins as well. But this is the normal pattern of initial infection of HIV. And so the CCR5 gene is, in fact, a very reasonable candidate for resistance to HIV. And it's been found there was a frameshift mutation that's found in many populations that greatly alters this protein. And in fact, people bearing this frameshift mutation are highly resistant to HIV infection. Now, there's been many other loci that have been involved. Here's a gene that codes for a chemokine that also affects this receptor usage. And there's been studies showing that depending upon what your genotype is at this chemokine locus, here you can have, this is age of onset after HIV infection, and some people are very resistant, others develop AIDS very rapidly. Also, these interact with the other loci, so that you can end up with individuals with combinations of genes that are completely resistant to HIV, progressing to AIDS. And these are studies done in South Africa where the incidence of HIV is very high. If you look at the most resistant genotypes and translate how this affects their reproductive fitness, they have a fitness of about 0.8. On the other hand, the most resistant genotypes have a fitness of about 1.3. So in fact, we see very strong selective forces being induced right now in South Africa. And so the South African population is indeed evolving via natural selection and adapting to the HIV virus. And it's very powerful, very rapid natural selection going on. Now, another aspect of our culture is that the culture itself is an environment. And in many cases, it's not that it eliminates selection, it just changes the nature of selection. And to show this, we can go to one of the classic textbook examples, the sickle cell gene. So this is due to a mutation in the 6 codon of the hemoglobin beta chain locus. And so here is the sickle cell gene that now has a valine instead of a glutamic acid. And this has a whole variety of phenotypes. I don't think with this audience I need to go through this, all the phenotypes associated. But from the evolutionary point of view, these last 2 phenotypes are the important ones. What is your viability in a non-malarial region? Okay, it turns out the individuals who are homozygous for the A allele have high viability. The heterozygotes have high viability. But individuals who are homozygous for the S allele have sickle cell anemia and have very low viability. On the other hand, viability in a malarial region, it turns out the AA have low viability because they're susceptible to malaria. The heterozygotes have high viability because they're resistant to malaria. And once again, the S homozygotes have low viability because of anemia. Now, most deaths occur due to anemia or malaria occur before adulthood. And so this can have a very big impact on natural selection. And what is important, as you can see here, is in a non-malarial environment, the S allele is a recessive allele for a genetic disease. But in a malarial environment, it's what we call an overdominant allele for resistance to an infectious disease, in this case, malaria. So when we use our culture to drain a swamp, to make an area that was a malarial area no longer a malarial area, we're not eliminating natural selection on sickle cell, we're simply changing it. We're changing sickle cell from an overdominant allele to a recessive genetic disease, but there still is natural selection. And this selection can interact with other evolutionary forces such as gene flow. So for example, when we look at African Americans, this was a study measuring the degree of admixture in African Americans, and they had many loci all showing about an 11% admixture rate, so that this particular African American population was 89% European in its genetic, or African in its origin, 11% European. But interestingly, there were several alleles all involved in adaptation to malaria, and look at their M's. They're much, much higher. Why? Because now there was selection for the European allele, that is the allele that is not the sickle cell allele in these cases. And so the gene flow was being amplified by natural selection because now these African Americans were in an environment that no longer had malaria as part of it. So this is the other theme we see. This increased genetic variation due to gene flow actually amplifies our responsiveness to natural selection. And here's a very dramatic evolutionary process that has occurred just in the last 2 centuries in our species, in this population. Now, our culture also not only alters, but it can also create novel selective forces that shape adaptive human evolution. And in fact, we can look at the malaria story in a little bit more detail, and we can add another player to it, the hemoglobin C allele. It's in the same codon as the sickle cell allele, but it goes to a different amino acid. And when you add the C allele onto it, you now have 6 genotypes, and these are actually the empirical viabilities in Western Africa in a malarial region. And you can see there's a lot of differences among these genotypes. Interestingly, the CC homozygote has the greatest resistance to malaria, much greater than even sickle cell heterozygotes. Now, how do we get malaria in wet tropical Africa? In fact, it was due to culture. About 2,000 years ago, there was a Malayo-Indonesian colony established in Madagascar. And in fact, even today, the language of Madagascar is affiliated not with Africa, but with Indonesia. In fact, at this period of time, the largest empire in the world was the Javanese Empire, not the Roman Empire that was so critical here. Of course, nobody knows about it because world history is a joke, at least in my country. I don't know how many of you knew about the Javanese Empire. That's what I thought. Okay. Anyway, this empire was very important because the colony brought with it an agricultural system. And was a very different agricultural system than the system that evolved here in the Middle East, which is a grain-based agricultural system based on annual plants. This was a perennial-based plant system, often called the root fruit. So bananas, yams, taro, that came from this agricultural system, and it used plants that were very good in a wet tropical environment, where grain-based agriculture is not. And this agricultural system was taken up by Bantu-speaking peoples, and there was a big expansion of the Bantus throughout much of wet tropical Africa about 1,500 years ago, and they brought this agricultural system with them. Now, this agricultural system is associated with slash-and-burn agriculture. This was important because this provided the breeding sites for Anopheles gambia, the mosquito vector for falciparum malaria. Even today, if you go into the areas of the wet tropical African rainforest that have not been cut, you don't find malaria there. And the reason is you don't have this mosquito vector. So this agricultural system of humans changed the environment in favor of this mosquito vector. And moreover, an agricultural system results in high local densities of humans, and particularly this one does, because when you plant trees, you have to stay put. Okay, you can't sow your wild oats like you do around here and then move on. And as a result, you have the conditions that allow malaria to become a very common epidemic disease and sustainably epidemic. And that created the selective forces that resulted in sickle cell going to such high frequency. So in fact, sickle cell is a cultural adaptation. And everybody gives maps showing sickle cell and malaria. Here I can show you the agricultural complex and the sickle cell trait. Sickle cell is really very much an adaptation to a particular agricultural system. Now, the interesting thing is that hemoglobin C in all of this. Here we see the genotypes associated with C, the heterozygote, and then the normal so-called homozygote. And one of the things you see is that unlike sickle cell, which has its resistance in the heterozygote form, C is only resistant in the homozygote form, and in fact the heterozygote is indistinguishable in terms of fitness from the normal homozygote. They both are susceptible to malaria. And without going through all the math, when you are favoring a recessive disease or a recessive beneficial trait, it takes it a long time. And you can calculate that it would take at least another 15,000 years for hemoglobin C to really spread in Africa. And it's only been about 1,500 years that we've had this cultural change, this cultural environment that caused the favoring of alleles for resistance. And in fact, we can just see the inklings of this ongoing evolutionary process. If you look in Africa, the C allele arose in Upper Volta, and it's gradually spreading out. And in fact, this is quite consistent with about a 1,500-year history of selection for this allele. But so here we're seeing an evolutionary process in the act. This is going on right now. Humans are adapting to malaria, here with a better adaptation than sickle cell anemia, but it's going to take a long time for this allele to spread. Will the environment stay a malarial environment all that time? I don't know. It could change and this selective process would stop. But right now, in fact, malaria is still a major selective force in this part of Africa. And so once again, we see that evolution has not stopped. It is proceeding right now. Humans are adapting to their environment. So I can now answer the question, and it's no. Human evolution has not stopped. In fact, it's in a very dynamic stage. We're completely restructuring the patterning of genetic variation in our species. And this is going to have all sorts of consequences, some of which we can foresee, some of which we probably can't foresee. Also, our culture doesn't eliminate natural selection, it just simply redirects it. And we see very powerful evidence of natural selection occurring in humans today that affect many diseases. And moreover, we do find that even the past natural selection that occurred can still have many implications. So if you want to ask, why do we have such a predisposition to hypertension, to diabetes, to coronary artery disease, to Alzheimer's disease? The answer again and again and again is evolution. So in fact, evolution, I think, is very relevant to medicine, both in terms of ongoing processes and understanding the etiology of the common diseases today. Thank you.